In this session, I am going to talk about recombinant selection and screening. It's actually an extension of third unit where we talked about the plasmid and vectors. So when we are using each type of vectors, there will be some mechanism or some provisions for selecting or finding the recombinant cells. So as we know that the frequency of recombination or frequency of gene transfer is very less. The maximum frequency in most cases is 1 is to 1000. If you are using 1000 cells, only one of them will be taking up the DNA. So we need to find the cell which is actually take up the DNA. So we are using selection and screening methods. And depending on the type of organism and type of vector we are using, the selection and screening methods will differ. So selection and screening has different meanings. When we use selection technique, we are using some sort of selection pressure, just like we are using the antibiotic in the medium. If you take the example of PUC80, PUC80 is having an ampicillin resistant gene in it. So if the cell has taken up the plasmid, or we are able to transfer the plasmid from the test tube into the bacteria which is receiving the plasmid will have the ampicillin resistance. And the non-transformed cells, the cells which do not take up the plasmid, will be sensitive to ampicillin. So that's the process of selection. We are using a selective pressure in the medium in the form of antibiotic. Second is screening. Screening is also a selection method. It can be classified under selection. But in screening, we are actually sorting the cell based on some features. Maybe based on the color, what we are using for the laxi gene. We have the blue cells and the blue colonies and white colonies, and blue colonies will be non-recombinant but has the plasmid, and the white one is actually the recombinant one. So like that, we can screen for recombinant cells or gene transfer, or we can go for selection. And both of these things, selection and screening, based on the expression of a particular trait. For the selection, we can classify whole selection methods into two: direct methods and indirect methods. And the selection methods also applicable for selectable markers as well as for screenable markers. So direct selection and indirect selection. There are different direct selection method. In the direct selection methods, we are actually looking for the function of a particular gene. Our plasmid will have a gene, and this will be expressed in the cell which is actually taken up the DNA. So if it has an antibiotic resistant gene, it will be expressed by the host organism once it get the particular plasmid. Or it has a laxi gene, it will express that protein. So based on the expression of a marker gene, we are selecting the particular cell. That type of selection is called a direct selection. Direct selection, inside direct selection, there are three different strategies. We can either use the marker or reporter gene, or we can go for complementation, and there is marker inactivation techniques too. So in the use of marker or reporter gene, our plasmid will have a particular gene, and based on the function of this gene, we can actually find which cell is actually transformed and which one is not. So maybe the best example is antibiotic resistant gene within PUC18 or PBR322. The cells which is actually taken up the DNA or the plasmid will have that particular trait. The Escherichia coli we are using for the transformation experiment will be sensitive to ampicillin. But once if it take up the PUC80 plasmid, it will become resistant to the ampicillin. So we can easily plate in two different plates, or maybe one plate. One is with antibiotic and without antibiotic. So one with antibiotic, only the transformed cells can grow, but the one without antibiotic, whole type of cells can grow. So if you want to have the cells which is actually taken up the DNA, add the particular antibiotic into the medium. That's all. And it also have some scorable markers. The scorable markers, the peculiarity is that we can actually find the intensity of expression, or how much recombination has been done, and it is a visual representation. So example for such type of markers are GUS, that is beta glucuronidase, and GFP. GFP is a very famous protein, green fluorescent protein. Once the cell is having that particular protein, the cells will glow in presence of UV light. So it's like we can find the recombinant cells or recombinant organism by using the UV light. So that is GFP. Second method is selection by complementation or nonsense suppression. This is what is working for especially the yeast vectors. You have seen the vectors like URA3. The yeast cells are auxotrophic for uracil, but the vector is having the uracil gene. So once the organism get the uracil gene, it can actually have the ability to grow in a medium without uracil. So it's like complementation. So, and this complementation can be also done in form of nonsense suppression, like forming a new type of RNA. So this particular type of RNA, this tRNA, will be complemented. Next, we have marker inactivation. I am not going to talk very much detail about it because we know the insertion inactivation technique, which is used in PBR322 and PUC80. In case of PBR322, if you are inserting your gene into an ampicillin gene, its ampicillin resistant gene, its ampicillin resistance will be lost. So the resulting cell will become actually sensitive to ampicillin even if it gets the plasmid. And in case of PUC18, it has LAXI gene. If you are inserting something to LAXI gene, the LAXI protein, the beta galactosidase do not form, and the cell will be blue in color. Okay, uh, actually, sorry, white in color. If we are inserting something into the LAXI gene, the cell will become white in color because it is not producing the functional beta galactosidase gene. So these are the markers or marker inactivation technique. Then we have indirect methods. In the indirect methods of selection, we are not selecting based on the function or function of a marker gene. We are actually selecting based on the DNA sequence. We actually want to find the DNA sequence or the RNA sequence or the protein sequence of the foreign gene, not the marker gene. So in this case, we don't really have any markers, but we are relying on the capacity of DNA, identification of DNA, or we are identifying the proteins which is produced from the foreign DNA. So it also includes three different methods, sub-methods: restriction enzyme cleavage pattern, hybridization techniques, and detection of specific proteins. So in the restriction enzyme cleavage pattern, after the transformation, we don't have any markers in here. So the thing is that in sometimes we could not use the markers. If you are actually doing the experiment for a commercial purpose, or we are using it for a creating a genetically modified organism, we cannot actually put a antibiotic resistant gene within it, because if you are putting an antibiotic resistant gene, it will be leaked out, or maybe if our plants is having an antibiotic resistant gene, it may be transferred to the bacteria, or bacteria may be taking it up and the bacteria will become resistant, and due to the conjugation capacity it can distribute this particular DNA. So for a large scale application, we cannot use the antibiotic resistant gene. So in that case, what we are using is restriction enzyme cleavage pattern. After transformation, we will be selecting all cells and we are isolating DNA, and we run a agar-cell electrophoresis. And before that, we will be doing a restriction enzyme digestion to shorten up the DNA. So if our cell is actually taken up the DNA, we can actually see an extra band during the electrophoresis. Okay, if the cell do not have that particular DNA, they do not have an extra band. It is a bit tedious task, but we can do it. So we are isolating the DNA, cutting with the restriction enzyme, running a gel, and we are transferring it to a paper, and we are looking for the DNA. So this technique is called bloating, and I will be giving seminars on this topic. Next is hybridization techniques. There are two different classes of hybridization: colony and plaque hybridization and bloating techniques. In the colony and plaque hybridization, what we are doing after gene transfer, we will plating it in a cell, and one plate will be kept as a master plate, another will go for the like something like a replica plating. Then in the replica, from the replica, we will put a nitrocellulose paper, and the cells will be transferred to the nitrocellulose paper. And in the nitrocellulose paper, we will do a mini miniature form of DNA isolation, and we will actually fix this DNA in a particular position. By using particular probes, we will actually find where our genes are. So this is called a colony hybridization. The same thing can be done for the plaques. In that case, we call it as a plaque hybridization. Instead of identifying the bacteria, we are isolating the plaques. Then we have bloating, like southern bloating, western bloating, and northern bloating can be used for identification of the particular gene or particular DNA or the RNA or the protein which is produced from the recombinant cells. And uh, we'll be discussing it more during the seminar session. We have combinations of this, like southwestern bloating, which can be used to find the proteins which is actually binding to the DNA. Next, we have detection of specific proteins. In this case, our foreign DNA or gene of interest we are actually inserting to the plasmid or to the vector is having some function, or some way we can actually identify this particular gene. For that, we can either use protein synthesis in mini cells or immunological methods. In mini cells, I think some of you may not be that much familiar with the mini cells. Mini cells are due to the abnormal division of bacterial cells. Here you can see the division should be in the middle, and each of the cells, daughter cells, should get the DNA and the plasmid. But sometimes the division will occur at one side. So one cell will be getting the genomic DNA, other will do not get any genomic DNA, but they get the plasmid DNA. So the one which got only the plasmid DNA, not the genomic DNA, cannot actually survive. It can live for some time, and it do not have the genome, so it will eventually die. But during the time of its life, it can express the proteins within this particular plasmid. So if this plasmid is having some function, this mini cell express. There are techniques to isolate mini cells, and we can transfer the DNA into the mini cell, and protein synthesized by the mini cells is exclusively from the plasmid DNA, not from the genomic DNA, because the mini cells having only the plasmids, they do not have the genomic DNA. So such that way we can actually find the protein function. And finally, we have the immunological screening. Just like we are doing the ELISA and all, we can actually transfer this colonies into a nitrocellulose paper. We can fix it. Then we can use the antibodies, which is complementary to the particular protein sequence, which is produced by the foreign DNA, and we can identify the colonies which is actually producing the particular protein. And there are methods which is actually used for screening by function. It has very limited application because most of these genes do not have any visible or clear function in the cell. So if the host cell is having some biochemical function, absence of some biochemical function, the foreign DNA will be able to complement that. If the foreign DNA is complementing that, that's screening by function. If a vector DNA is doing that, that is not this type of complementation. Then we can use specifically disabled functional hosts. So after the recombination, the function will be regained, or some of the cells after inserting particular DNA, they'll have a different function, just like GFP. If you're inserting a GFP protein, green fluorescent protein DNA into the cell, it will produce the green fluorescent protein and it will glow. Like that, we can use screen the cells based on function, but it has a very limited applications. The next topic I would like to talk about or mention is phage display. It is not directly in your syllabus, I guess, but I think it is better to know. It is discovered by George P. Smith, 1985. Phage display is a technology that allows the expression of a particular gene in a phage. So phage will be expressing this protein as a fusion protein. So fusion protein, some part will be from the virus and some part will be our foreign DNA. So this is a phage display system work. You are inserting particular gene into the phage genome. So it's a small part. The phage will be expressing its whole protein along with our particular protein. If you are inserting a immunoglobulin gene into the vector DNA, it will produce immunoglobulin gene as a fusion protein. It is fused with the viral protein. Or if you are inserting insulin DNA to this one, it will be expressing insulin as a fusion protein. Okay, fusion protein is the combination of two different proteins. So most commonly used vector for this fusion protein production is M13 vectors. M13 vectors is having two different proteins, P8 and P3. We can actually make fusion proteins with the P8 or P3, and it can be used to study protein-protein interaction, protein-peptide interaction, and the protein-DNA interaction. And it's a very useful system for finding the recombinants. So that's all for this unit. Thank you so much.